I'd like to thank everyone for joining today's webinar. We are very excited to host Dr. Neil Paragas from In Vivo Analytics, who will be presenting on their quantitative 3D imaging solution. Dr. Paragas earned his PhD in biology with distinction from Columbia University, focused on modeling human disease in small animals using bioluminescence, animal genetics, and surgical methods. He has served in faculty positions at Columbia University and maintains a faculty appointment at the University of Washington. In 2015, Dr. Paragas co-founded In Vivo Analytics and introduced a first-in-class preclinical R&D analysis platform powered by artificial intelligence. With his firsthand experience in bioimaging, Dr. Paragas identified researchers' unmet need for an automated and quantitative, non-destructive bioluminescent 3D method, thus launching In Vivo Plot. Validated in high-impact publications such as Nature and Cell, In Vivo Plot represents the first quantitative bioluminescent preclinical imaging method to operate in the cloud environment. So with that, we'd like to extend a warm welcome to Dr. Paragas and look forward to the presentation.
Thank you, Melanie. Again, my name is Neil Paragas, and I'm really thrilled to be here today. And I thank everybody for inviting me. I'm co-founder and CEO of In Vivo Analytics. In Vivo Analytics is a preclinical R&D software as a service, image and data analysis company with a focus on small animal in vivo imaging. Today, I'm going to discuss a new paradigm in preclinical data analysis, In Vivo AX. This easily adoptable solution enables automated image analysis and is future compatible with AI tools. In Vivo AX is a powerful software that'll provide the end user with an automated data analysis tool that you can yield insights not accessible with conventional techniques. But today I'm going to speak about our lead commercial quantitative bioluminescent system, In Vivo Plot. So the biggest problem in preclinical imaging analysis is it's us. Cognitive bias is just part of us humans. And as we interrogate the data, these biases arise from numerous reasons, such as information processing shortcuts, noisy information. This is always a funny one. The brain's limited information processing capacity, emotional, moral, motivation, social influence. These are problems when it comes to the operator, which is us, interrogating the data, which is our job. So unfortunately, regardless of how sophisticated the software we use is, it's still operator dependent. It still requires an operator within the loop, within the circuit. So since the operator is within this investigative loop, they bring cognitive bias, variability, and an absence of reproducibility. And just in short, it's really image analysis is the unmet need here. It's not throughput. It's not the quality of the imaging system. The problem really lies in us, and we just don't have the tools to unlock the data. So 2 different people, 2 different results, 10 different people, 10 different results. And then once you get these data, let's say you do a good job, you're still putting these data into software that really wasn't designed to display it, such as Excel. It's really designed for business. There's a huge opportunity here. As we know, in vivo imaging, in particular optical imaging, has been around for decades. We collect terabytes of data, but we can't integrate all these data we're collecting because there's an absence of a common framework. There's an absence of standardization. So you can't include any data collected anywhere. And because of this absence of a common framework, it makes incorporating technology, which we all hear about every day, challenging, if not impossible, such as incorporating artificial intelligence. And without being able to achieve these things, the data is going to be as it is. It's not going to be as actionable, quantitative, or even shareable. Right now, we're really looking at an opportunity to make data analysis operator independent and unbiased, high throughput, enabling real-time data analysis to increase the efficiency of the workflow. And a number of studies have shown this, and the NIH has made it a priority to increase reproducibility. So as I just mentioned earlier, there's a real problem because of the absence of a common framework. And when you're imaging animals, such with unknown animal size and pose, that prohibits automated data analysis and automated processing, image processing. And the underlying problem is incongruent data. So to solve this problem, we need to force congruency. Without a defined structure, imaging animals at different size and positions leads to incongruent data. Whereas defining the animals in a grid-based system leads to congruency. So how do we do this? The state-of-the-art analysis is primarily operator-driven, as I mentioned earlier, who manually connects data points of different animals. And since the animals are in different poses and positions and have different sizes as well, the manual data connection is time-consuming, operator-dependent, and subjective, and prone to error. Using our technology, all animals are defined on a regular grid, which provides a uniform structure across different animals to enable data congruency. Data congruency is where all the data points correspond to one another, and they provide the required foundation for machine-driven and automated data analysis. So the first solution to this problem of an absence of a common framework is our patented body-conforming animal mold that forces the animal in this defined position, a known geometric shape. Being in a defined position, we can embed an organ probability map, which is our patented digital mouse atlas. Current imaging techniques prohibit automated ROI alignment across different animals because of this missing spatial data point congruency between animals that are different sizes or different shapes. Therefore, manual ROI selection is it's currently necessary, which is time-consuming, operator-dependent, and prone to error. The body-conforming animal mold, or the BCAM, enables data congruency and hence provides this uniform spatial framework for co-registration across different animals, time points, and even imaging modalities. This work was validated by our group in Nature Communications. And I'm happy to provide the link later. The body-conforming animal mold is scaled based on animal weight. We have a wide range of molds in 2-gram increments available. The body-conforming animal mold is made of an optically clear polycarbonate macrolon, which is a clinical grade plastic. It can be cleaned with bleach or ethanol, can even be autoclaved if necessary. It's also can be disposable. Based on the size here, it creates this uniform framework. And you can see, regardless of the animal size, when imaged with a CT, you're getting a very consistent alignment. And this is kind of a subjective analysis right here, but your eyes don't lie when you can see the spine. Additionally, the BCAM provides anesthesia delivery and gas scavenging, and that's all incorporated up in this head unit, which plugs into the imagers. This allows the anesthesia not to pool. It also allows more efficient gas scavenging for the operator. The other problem in optical imaging, especially if you want to be quantitative optical imaging, is in light transmission. And the BCAM solves this problem because we're going to know where the animal is in space. If you see here, here's a mouse with light being transmitted through it. And you see blue, which means a lot of light is being absorbed, and these red, light is being transmitted through the subject. And there's just a wide range of light transmission properties, tissue properties in subjects in mice, which is what we typically use. And this complicates the data analysis. And how do we address this? The first way we address it is the organ probability map, which is a statistical anatomical atlas. It's aligned to the body conforming animal mold, regardless of which mold we pick, whether it's a 14 gram, a 36 gram body conforming animal mold, the organ probability map will scale to fit within it. It primarily, for image reconstruction, allows us to know the different tissue types spatially to build matrices, along with determining where partial blood volumes are. Additionally, on the analysis side, it then guides users either with for an automated ROI delineation without the need for contrast, or it tells the user what region their signal might be in, and we'll get to more of that later. There's many data points in the BCAM, and this allows very accurate alignment amongst all the subjects. Instantaneous anatomical reference is what this offers. And unlike other atlases, this is a statistical anatomical atlas, meaning many mice were used to build this organ cloud. So it's a probabilistic map of where a tissue type might lie in the space within the BCAM. We know this works, and we know this works well. Again, in our Nature Communications publication, we compared the Dice coefficient to all, at that point, all the other atlases available. And InVivoPlot, regardless of gender and animal strain, had a better registration accuracy than all the other ones, especially in tough organs, such as the spleen. Mean surface distance was also very strong, along with VRC, recovered organ volume. And again, you can find these data with more thorough descriptions in our paper. The BCAM and the organ probability map operate within our cloud-based software. We're using software as the real solution here. So InVivo-AX is a preclinical R&D software as a service that I mentioned earlier. Image data is uploaded, whether it's from InVivoPlot, which is what we're to speak about today, which is quantitative bioluminescent tomography, or our other optical imaging tools. So the mouse shuttle really enables us to automatically do image processing, whether it's coming from, let's say, nuclear imaging, and here is a Molecube's PET CT, or it's coming from optical imaging, which is what we're going to talk about today, and here's the SII logo. Image data is acquired on these units and then uploaded to the cloud, where it's processed, put into databases, and once within the database, we can automate the study results and iterate. The data can be shared, it can be reused. It doesn't need to be continually processed. So let's dive into InVivoPlot. So InVivoPlot is the quantitative bioluminescent tomography tool. And it addresses really the problem, which I mentioned earlier, with the light transmission problem for quantitation. Sure, there's other methods for bioluminescent reconstruction to make a three-dimensional image, but those other methods are not truly quantifiable because they don't treat the subject as a heterogeneous organism. They treat it as a homogeneous organism. So where we treat every voxel as a different cell type, we treat every voxel with a different blood volume. The other solutions out there treat it as a mixture of muscle and fat, a homogeneous mixture of muscle and fat. So if we take two equally bright capsules, and these capsules contain a bioluminescent bacteria, and we surgically implant them below the kidney and below the bladder, and we image those using a mirror gantry, so simultaneously imaging the dorsal and ventral side, you don't need analysis software to tell you that there are different signals coming from these ROIs. So the solution is to utilize a mirror gantry along with the body conforming animal mold along with a pre-calculated high-order tissue matrices for the reconstruction parameters. And we did that with Spectral Instruments Imaging, using their logo, developed the mirror gantry, which holds the BCAM. It easily clips into the gas anesthesia, gas delivery, and gas scavenging ports on the Logos platform, and mounts with some pins in the back that are on all the Logos. And then images are acquired. Here we're going to get a multi-view image. And to do the reconstruction, we need a multi-view, multi-spectral image. And I'll get to that in some of my later examples. But first in the validation study, and this is still all in the Nature Communications paper that everybody can refer to, we take a kidney infection. Here it's bacteria that is uropathogenic. It ascends up into the kidneys. And here we have a kidney infection. And the question was, can we quantify, can we count the number of bacteria, of number of colony-forming units inside the kidney? And the answer is yes. By using in vivo plot, we can reconstruct this bioluminescent image and align it to the organ probability map and quantify it. And these data show that in vivo plot has a very high, a very good ability to quantify in vivo the amount of colony-forming units compared to ex vivo. And ex vivo, you take the kidneys out, you homogenize them and plate them and count the cells. Manual ROI, meaning you're going to draw an ROI either on the dorsal or ventral side, and you don't get the same ability to quantify the number of bacteria in the kidneys. Another example of using our technology, and here I'm going to start walking you through the software, because we have a bioluminescent mouse, whether it's infectious disease, it's cancer, stem cell, T cell, whatnot. You're putting these bioluminescent reporter cells into the subject, monitoring them over time. You're wanting to watch them, whether they're going to grow, whether they're going to shrink in size. But how do you quantify it? Typically, you're going to be acquiring the image on the machine, going into the software, drawing ROIs, and then putting it into Excel. Here, it's a little different with in vivo plot. So this is the osteomyelitis model. This is a bone infection model that we did with Frank Lowy at Columbia University. So this is the way the software looks. You log in, and just scoot back. You log in through this web portal. Once you log in, I'm going to pause here again. You're going to be you're going to see the opening page, which is your projects. And in the project page, you can either pick one of your projects, you can create a new project. Here, we've already made the project with Dr. Lowy, a bacterial infection. In this window, we're seeing the number of cohorts that are available, or that we created. You can have one cohort. If you're just looking at one group, you can have multiple cohorts, unlimited number of cohorts. And within each cohort, you can have a single animal and that subject. And then that subject is then further divided into time points. You can see here, there's different animals with different time points. So this is the osteomyelitis model. This is a bone infection in the femur. The femurs were opened up and these bacteria were placed in them. So here we selected a single animal. This is animal number 65, day 4, cohort 1, and it's in the project, this Dr. Lowy's bacterial infection project. So right now we're visualizing the single animal, single time point. This is the slice view, and what's just was loading is the co-registered anatomical atlas. So we can see here, this is here's the mouse. This is reconstructed in slices. You're in 1 millimeter slices, coronal, sagittal, and transverse slices, and the corresponding organ probability map that you can select either all of the organs to be aligned with your image, or just one of them. Here we have the skeleton that I showed you, clicking back and forth, going back to Dr. Lowy's project. Again, ROI selection. That first ROI I made, well, I haven't made an ROI, I guess yet. And so now we're adding a project ROI. That's the whole animal. We're just going through. And so when I'm adding these ROIs, what's the powerful thing is that it's all 16 of these subjects. So when you're visualizing here, this is the speed. This is a pretty simple case where it's just taking all the ROIs from the whole animal and porting it over to the visualization software. We're going back to cohorts, and then within cohorts, we're going to click again into cohort 1, and I think I'm going to be making another ROI. See, in vivo plot. And at this level, you can see everything's hierarchical in the system. So this is the project, this is the cohort. So now I'm going to add a cohort ROI. And the cohort ROI can do the same thing, add cohort ROI, and it'll be labeled as such. So if you have multiple cohorts, you could do a whole animal for cohort 1, a whole animal for cohort 2, the whole animal for the whole project. Or you could dive down deeper and do that for each individual organ group. So moving forward, this is how the data is displayed. You can change the chart type, maximum, median, mean, linear, total, volume. There's many different ways to interrogate the data. I'm just clicking through, just move forward, doing some more ROI selection here. Now I'm adding the skeleton as an ROI. You can see the skeleton coming up. So this is going to be at the project level. So now we have the skeleton and the whole animal at the project level. You can see these two hotspots here in this coronal slice. I'm going to create a manual ROI now. And I'm going to call this one right femur. And when you create a manual ROI, your ROI group comes up here, and then we can start editing. And I'm going to pause really quickly here. If you notice these white dots are individual peaks from all the subjects. So 16 animals. So in this cohort, we have 16 subjects. All 16 subjects are being interrogated together. Their data is aligned. So we're looking at the average of all 16 to draw these ROIs. And what's really nice about the VCAM is you're already noticing that the alignment of each slice is very accurate by each one of these dots, these peak finder dots, showing that at that slice, coronal, sagittal, and transverse section, you're having the same peaks. And you can use that in with or without the organ probability map, really to guide your image analysis. So you see here, I'm just clicking away. The ROI tool is a volume tool. So this is a sphere. You're drawing a sphere in that region. And there's different size spheres, and then there's different erasers. So it's another sphere that will erase the ROI you're drawing. Right there, I saved that ROI, and now it looks like I'm going to make another ROI. I'm going to call this one left femur. And in left femur, I'm going to do what I did on the right side. I'm going to go through, I'm going to edit, and then I'm just going to start drawing the ROI. So again, what this is doing is I've effectively drawn 2 ROIs on 16 different subjects in 3 dimensions. And as we continue to add data to this project, it'll automatically align these data and incorporate them into the charting, which is in real time. looks like I'm just showing you that you can turn these ROIs on and off. And then now we're going to the analysis that says in vivo plot. So you click over, and to be expected, it says statistics are still being calculated. This is a big data set. So you can refresh or just wait a minute. And if we refresh the screen, there's our data. And this is all happening. So you have to think, you have this 100,000+ data point subject that you have 16 of them, and they're being analyzed simultaneously. And here, you can see here's the right and left femur. And this is going to be in the source density. Let me keep moving because we have a lot of other examples. We're still in infectious disease. So recently, Dr. Duam at the Needle at Boston University approached us and said, can we do biodistribution for a SARS-CoV-2 model? And we were thrilled to be able to participate in this. And they recently published this in Cell Reports. And they basically implanted 2 human lung xenografts on the back of a mouse, infected one, and wanted to see whether the SARS-CoV-2 would go from one xenograft to the other. And they did not see that, which was a powerful piece of data, along with the GIF MIP image that they were able to output. The GIF being just a graphical image type that you can use for presentations, and MIP is a maximum image projection. That's the 3D output. Dr. Crossland, also at the Needle, was investigating SARS-CoV-2 infection in a little bit of a different model, but they were interested in how these SARS-CoV-2 infections would transit in these specific type of mice. And they were excited to see that post-infection, that the SARS-CoV-2 infection would then transit up to the brain. And here, there's many different ways to utilize the software. So here you see a view that they took from the MIP image. Okay, moving on. Moving on to immunology and immuno-oncology. So some work that we did with Juno Therapeutics for a safety study. So typically in immuno-oncology and T-cell therapy, to know where the T-cell goes outside of just watching the tumor shrink, the gold standard is histopathology, which is ultimately pretty limited when you're dealing with genetically modified T-cells. So Juno was interested in seeing if they could monitor these genetically modified T-cells in vivo longitudinally and quantify them with a biodistribution type technique. So what we were able to do, and here's just an overview of the data, and I'll dive into what the data looked like before and after in vivo plot. They were able to upload their data, analyze it on our cloud-based platform. They interrogated it. They had 50+ time points that they looked at. Isolate where the signal is. Do image reconstruction here, and you can see with and without chemical stimulation, they see the T cells trafficking, which is exactly what they wanted to see. And then also quantification of these trafficked T cells. So their problem really is they came to us and they said, we don't really know where this signal is. You can't do a biodistribution study not knowing exactly where it is. Sure, you can open up the animal and do some ex vivo imaging of the organs, but that's not truly quantitative either. Or you could do histopathology But there's a lot lost in that technique as well. And so you get this multi-view image, as I mentioned earlier. So we take a multi-view image with the mirror gantry. And something I didn't mention earlier is the technology depends on the process of taking a multi-view, multispectral image. So here is a complete data set, a multi-view, multispectral image. Each subject is cropped out and processed. It's surface mapped onto the surface of the subject. And we take a 360-degree view here. And the importance of the 360-degree view is that there's no other way other than having a 4-camera imager, which really don't commercially exist, to get this image. So this is really the only solution to take a 360-degree view at a single point in time. And then you have to take that view with filters, spectral imaging, and open filters to understand the kinetics, the luciferase kinetics, while you're collecting these images. The only other commercially available tomographic tool out there collects just a couple spectral images, and it guides the user to collect it at the peak of the luciferase expression, so the plateau. You're gonna have to find the plateau and image at that time point. So if that's a half an hour after luciferin injection, you have to wait that time and then you acquire the image there. Needless to say, that's not very accurate because the plateau might change from subject to subject. And then finally, it's the data. So you take these data and upload these data to the cloud. In the cloud, the analyses and normalization is all baked in. So the user doesn't have to know about optical physics to really understand how to do this work. So back to immunology. This is one of our clients at Vanderbilt University Medical Center. And I thought they had a kind of an interesting project. And they were just interested in seeing where do these T cells go when they put them into the peritoneum of these mice. So they do an IP injection of T cells, and then they monitor it over time. A half an hour, a couple hours later, 8 hours later, watching these T cells home to the different regions. So the software can be used just for very straightforward purposes to understand where the signal or the cells might be migrating to. So this is some work we did with Phil Greenberg at the Fred Hutch, and Phil's group was also interested in T cells, and this is a pancreatic cancer model, and they wanted to ask with the different targeting between treated versus untreated T cells. And as you can see here, just in a planar image, it was very challenging for them to visualize where these T cells were going in relationship to the pancreatic region. And tomography really helped them get to that point, along with being able to quantify it. Here again, we're going to have another example of the software interface. Again, here's the project cards. In the project card, we can go to Dr. Greenberg's card, ROI selection. This is just grid. Here's plot. Grid is our planar tool. There's whole animal. You can see here is a signal. The system defaults to grayscale, and you can always switch to pseudo color. Again, clicking through the different ROIs. These, if it says OPM, that means it's the organ probability map ROI. This pancreas was a custom ROI. This is an ROI we drew ourselves. Spleen, we just added spleen to this project. It's taking a second to load. It's loaded now. And then now you can see the spleen. This is another medium-sized set of 17 subjects. So here, again, I'm going to point out all these white dots. these white dots indicate peaks. These are all peaks from the 17 different subjects in this cohort. Just clicking through, we can adjust the sensitivity of the peaks. And now we're going to analysis. So we created a new ROI, and then we can go to analysis. You can chart the analysis however you want. We have different ways of looking at the data: maximum, mean, median, volume. You can change the views, the axes views, logarithmic or linear. You can upload files here. I'm not going to show you today. Export project data into tab delineated files, CSV files. Okay, I'm continuing to interrogate. I might just fast forward a little bit. Here I'm creating another ROI. And I'm calling this ROI random. It's just in a random location. And we're just going to, again, see the kind of the speed of the reconstruction, and also the accuracy. We would anticipate that there was low signal in that region because we can't see it here. So it's calculating it, and there it is. So we have, I'll pause here for a second. We have 2 cohorts, 1 and 2, and we have a number of different ROIs visible. And we can change these axes. It looks like I'm going to change the axes to time. And these are the 4 ROIs. Over time, we can toggle them on and off. Again, a lot of flexibility in what you view. If you like this chart, you could save the figure, or you could take a screenshot. You can always, again, export the data for the graphical software of your choice, like a GraphPad Prism, for example. And we continue to interrogate the data. And let's just keep moving forward a little bit. So again, lots of different ways to look at it. And so now we're going to go into an individual animal. And again, everything's hierarchical. So we have the project, the cohort, animal, and then the time point here. We're going to the volume view. There's surface view. So surface view is the different surface mapped views from the different filter sets. And those filter sets is what the software uses for the image reconstruction. Here in the volume view is the slicer. Here we have the slice view with the coronal, sagittal, and transverse slices that are aligned with the organ probability map. And here we're just turning all, well, I can see what I do here. Projects, so these are the project ROIs. And here's no ROIs, and then we can toggle around with the different OPMs. And just fast forward through here, and then different sliced views and whatnot. Okay, so the next cancer model I want to show you is a lung carcinoma model. And this is an interesting example in that it shows how can we use the organ probability map when the signal starts moving outside of where you would expect it, where it's statistically predicted to be. And this lung carcinoma model is a good one because even though these lung cancer cells do embed themselves into the tissue of the lung, they grow into the body, away from where the lung is. Again, here's the software. We're logging in. We're going to this project, which we called lung tumor. Here's in vivo plot. It's a smaller group. It's just 4 uploads. And so you can delete ROIs like I just did right there. And here I'm adding an ROI, and I'm going to call this ROI tumor. Okay, it's a tumor ROI. It's a creative one. So in tumor ROI, it's going to create this ROI, this custom ROI for us now to utilize. You're not seeing anything here because it's most likely in these slices that we're looking at. There's no signal. Let's just move around, navigating around. I'm going to pause here. This tool in the toolbar allows you to navigate through the sliced view. You know, there's a red bar here. You could go ventral to dorsal, and then here it's left to right, and then, you know, here anterior, posterior, and whatnot. Again, you're seeing these peaks. Much smaller data set, 4 animals. Regardless, you're still seeing alignment of all these animals in the same region. Reproducibility. So it's moving through here and then drawing the ROI based on the peaks that we're seeing. And this is a combination of all four subjects are in the same space. And then there we go. So, you know, a smaller data set, it's going to calculate the values much faster. But you can see right there that we get the tumor ROI here. It's very dense. There's a source density, so it's light to volume to time. That's what the output is. You can see here. Let me zip through here. You know, there's correlation tool. We can get at some other time. And this is what I was talking about earlier, is when we're looking at the signal here. Here's the lung, OPM. Here's the signal of the tumor, lung tumor, and then the OPM is helping guide us. And it seems like it's right off to the side of the lung. You see here, here's the right side, right down there. So this is where the lung tumor began to grow, just down on that lower right side. All right, let me keep moving. So here's another model. This is another immuno-oncology model. Doctors Lau and Desis at University of Washington. And again, they had a disseminating ovarian cancer model, and they wanted to see, to quantify these disseminating tumor cells because they were so dispersed. And since they're disseminating, it becomes very challenging to accurately quantify them. These cells will go, they'll stud the spleen, they'll go around the diaphragm, all over the place. So they're amongst and mixed in different light-absorbing tissues. Here's also work by Dr. Miyazaki at the University of Washington. He published this in Communications Biology. And he used the system to determine the spatial location of light-producing kidney cells. So this is bioluminescently driven luciferase kidney cells. And he aligned them with the organ probability map and was able to quantify their relative expression to one another. And that's really what I have to show you today. I really wanted to show you an overview of how in vivo plot can be used to reconstruct planar bioluminescent data. It can be used for very quantitative tool applications like biodistribution. It can give you spatial guidance, quantification. You can do 4D imaging, which is where you take a 3-dimensional image and you image that over time, and you can stitch those images together to tell a story of how, to tell a story of where these cells might be going with their immune cells, their stem cells. They can be bacteria, they can be viruses. They can also be drugs, small molecules. So there's a lot of applications for this tool, and I'd be thrilled to answer anybody's question about it, or please just follow up with us and we can organize a time to talk about it. Thanks for listening.